commentary on a paper I discussed last week talked about like the overall goal in synthesizing islatravir, and it kind of summed it up nicely here. So basically, unnatural nucleosides like reverse transcriptase inhibitor islatravir are generally challenging to synthesize. So a team was able to re-engineer a bacterial biosynthesis to run in reverse, kind of like highlighting their retro pathway that they planned out backwards to run in the proper direction. And they evolved 5 different enzymes, which we're going to call just the evolved enzymes. And these enzymes allowed the pathway to do what they needed. And then the 4 auxiliary enzymes drove the reaction in the direction they wanted. So just a review of the enzymes, the 5 evolved enzymes of the pathway. So this is the whole pathway. And then these colored enzymes are really just I don't want to say the most important, but like really just flashed ones, I guess, the ones that they just evolved from their wild type enzymes to do and trained to do what they wanted in the reaction. So this table highlighted, I talked about this last week, it highlighted all of the evolutions to the enzymes that they happen and the goal, but this visual kind of summarized it a little bit better. So for example, GoAce went through 12 evolutions, like 12 rounds of mutations, and its goal was to better the activity in the proper direction and do so in a stereoselective way. PAN-K mainly was evolved for better activity. DARA was evolved to withstand high amounts of aldehyde, and PNP and PPM were evolved to have better activity. So just to review these enzymes, GoAce was able to properly form the compound 7 over here and do so with the proper or proper chemoselectivity. So this alcohol here was turned into an aldehyde, and these had the proper direction. So this alcohol became, wedged, and this alkyne went back into the paper as a dash. The PAN-K basically just phosphorylated this compound 7, so it took this alcohol here and phosphorylated it. DARA was able to convert this aldehyde into this ribose here using this mechanism that I talked about. So what was interesting about DARA was that it was evolved from the wild type enzyme, and like I said, it was trained to withstand high concentrations of aldehyde, and this was impressive because aldehyde solutions are generally used to kill protein activity, so the fact that it could enhance and better the protein activity was really impressive. The PPM changed the location of this phosphate group, so I summarized from carbon 5 here to carbon 1. And then the PNP was able to bind this unnatural phospholipase and this unnatural fluorinated base between this nitrogen here and this carbon here, and make islatravir with this phosphate group just kind of floating around. And now the auxiliary enzymes were really important in this process as well. So basically the implications of the auxiliary enzymes was to take adjacent or unnecessary parts of the biocatalytic pathway of islatravir and use them for something productive and irreversible and able to ensure the efficient synthesis of islatravir. So the HRP helped to maintain the correct oxidation state of the GoAce, and only a small amount of HRP was needed to activate the GoAce, which was the evolved enzyme that this enzyme was paired with, and this made it pretty cost effective. And the paper that this fact was sourced from indicated that there was a 16-fold increase in the specific activity of a sample that used HRP as to catalyze the reaction versus those that didn't. So it made it pretty essential for the catalysis here. The bovine.
And can I just mention what that specifically means? That specific activity term. That's the amount of catalytic activity, like per amount of enzyme. So all that means is that the horseradish peroxidase, as an additive, let them use less GoAce than they would have had to have used if they didn't add the horseradish peroxidase. Yeah. All right.
Thank you. So now for the catalase, so H2O2 is a byproduct of the GOACE reaction right here. So all three of these are paired together. And what catalase does is it just catalyzes the decomposition of hydrogen peroxide into water and oxygen. For the ACK, so it's paired with PAN-K here, unlike this protein bead. So PAN-K was able to phosphorylate 7 right over here efficiently. So it took this alcohol group and phosphorylated it. And it did so like efficiently and consistently and in the correct stereo configuration. But doing this process in vitro or like in the test tube or culture dish required regeneration of the specific cofactor being used for the phosphorylation, and this cofactor was ATP. So this is where the ACK enzyme came in, and ACK used acetyl phosphate to regenerate ATP from the ADP byproduct in the second step. And then SP, which is paired over here in the last couple steps. So since the natural salvage pathway runs in the reverse direction, or like it's reversible for the ppm and PNP steps, SP was the thermodynamic driving force that drove the reaction to produce islatravir successfully. So the addition of SP to remove the inorganic phosphate that was like floating around up here, allowed the mixture to produce high concentrations of islatravir. So all of these enzymes were equally important in the mechanism. So just to review, these enzymes turn this alcohol into an aldehyde with good stereochem, the ideal stereochem. This alcohol became phosphorylated. The DARA helped to turn this into a ring in high concentrations of aldehyde because acetaldehyde was the what made this a ring. ppm was used to isomerize this phosphate from carbon 5 to carbon 1, and then the PNP was able to bind this group with the unnatural fluorinated base to make islatravir, and the SP removed the phosphate. So now we got to ask, like, what's islatravir? So just to refresh, so islatravir is an HIV investigational drug. So, but it's still in the investigational stage, but it's like pretty impressive in its synthesis and, you know, its use, and its target is reverse transcriptase. And now that we know that, kind of like have to think backwards, and we say like, what is reverse transcriptase and how does it work? So this video highlights it, but instead of playing the video, I just kind of did a rundown of it. So retroviruses use RNA rather than DNA to store its genetic information. And we know that DNA turning into RNA is transcription, but then we ask, like, how can RNA be copied in these retroviruses that use RNA as its genetic information? So reverse transcriptase is the enzyme that catalyzes reverse transcription, which is exactly what you think it'd be. So it's turning RNA into DNA. And this allows the RNA to be copied in human systems. So HIV uses reverse transcriptase to replicate and spread throughout the human body, which is not good. We don't want that. So knowing this, scientists were able to develop something to prohibit this from happening. So now we come to EFDA. So a more recent article talked about EFDA and how it was a pretty potent anti-HIV agent, and I was like, well, what about islatravir? Like, I thought this was pretty good. So EFDA is islatravir, just with a fancier, more scientific title, so they're the same thing. And it has pretty impressive pharmaceutical significance. So it has an unusually long half-life, which is good because in one dose it can kind of last longer compared to, I guess, other investigational drugs. And its mechanism of action is different from other previously approved anti-HIV drugs. So what it does is it suppresses the rate of viral replication by diminishing reverse transcriptase translocation, which means the RNA won't be really turned into a protein. It kind of stops it from getting that far. And it does this at the beginning just on the nucleic acid substrate. And it also can act as either a de facto immediate chain terminator or as a delayed chain terminator. So what does this mean? So chain termination is any chemical reaction that halts the formation of reactive intermediates in the chain propagation steps of polymerization. So it just kind of keeps it from polymerizing and becoming a bigger protein. So in other words, the HIV RNA would not be replicated in the human system if chain termination were to happen. So as it implies, the de facto, or in fact, in reality, intermediate chain terminator would stop the replication earlier rather than later compared to delayed chain terminator. And so I was thinking, well, obviously, like stopping it immediately would be better than delayed, right? But I found this in the paper. This is like a heavy sentence, but it said, finally, when EFDA acts as DCT, EFDA-MP terminated primers are protected from the phosphorylatic excision that leads to NRTI resistance. So that's a heavy statement, but I tried to break it down so I could understand it better. So EFDA acts as a DCT. This is saying when EFDA uses delayed chain termination, and then EFDA-MP terminated primers. This means EFDA monophosphate primers, and these could also be considered primers for islatravir that lack a phosphate group already, kind of how I interpreted it. And then protected from phosphorylatic excision. So phosphorylasis is similar to hydrolysis, where you cut or break something or cleave something using water. Phosphorylasis cleaves something using a phosphate group, and excision is the removal of. So I tried to use like word math here and determine that phosphorylatic excision is the removal of a phosphate group. So I guess this would be like reverse phosphorylasis.
It actually probably is talking about the removal of the islatravir.
Okay.
Yeah, so it's like when islatravir is in there, it's sort of like tricking the virus to use islatravir instead of a natural nucleoside.
Okay.
But there's also mechanisms that can, after it gets added, it can get cut back out so that the virus can like try again to make one that isn't like poisoned with islatravir.
Okay.
So the excision refers to removal of an incorporated molecule of EFDA or islatravir.
Okay.
Yeah.
Well, so protecting it from this happening, because that could lead to NRTI resistance, which means that islatravir wouldn't work against HIV. So I guess it's ideal when EFDA uses the delayed chain termination mechanism, because then it wouldn't pose the threat of being removed in the presence of HIV. Right, okay. And then just like a fun kind of fact, this parallels how entecavir, which is an antiviral treatment for hepatitis B viral infection, is protected from the same phosphorylatic excision, and it uses the DCT mechanism. And also the apparent mechanism of action for remdesivir, which we know is like a drug bouncing around in thoughts for COVID. That also uses chain termination in a similar way that entecavir and islatravir use it. So I thought that was pretty interesting.
That's true. I
Right.
do too. Yeah.
But yeah, that's all I have to say.
Yeah, I think, okay. Yeah, nice. Any questions for Emily?
Yeah, I have a question. So this is kind of like the beginning of this presentation, kind of like what you were talking about last presentation. So the retrosynthesis, is that just like, did they just call it that, or is that like an actual technique that's used to like plan out your synthesis?
Yeah, that's an actual technique that's used to like plan it out. Just like highlighting it, kind of working backwards.
Are any of you planning on taking DJ's advanced organic block
I think we all
course?
are.
I think we're all
Are
All
you?
of us.
Yeah.
Do you all ever make any independent decisions on your own?
That's actually really true.
Why
all
would
were
we
like,
do that?
I don't know. Yeah. I mean, at this point, it would seem silly to do that. I just curious. I mean, I actually might take that class too, because I think I could learn some organic chemistry sort of from DJ, because I'm almost self-taught, you know, so there's like some holes in my knowledge. But you will certainly learn in that class. You actually might spend a lot of time doing so-called retrosynthetic analysis, which is if you know what structure you want to make, how can you sort of plan a synthesis by going backwards? Like, so like what would be a final reaction you could do to produce the molecule you want? And then you have one step towards, you know, one step in then, and then can you do that again and again and again, and then disconnect all the way back to materials that you can like buy, you know what I mean? So that's what a retrosynthesis. It's hard to think about it in that way, but you'll probably get a lot of practice doing that in DJ's class. I think he'll probably spend at least a little bit of time doing that. But the one thing I wanted to talk about with this Letrivir too, which is like just a stunning little detail of how they did it, is that, you know, they had these things. They had this achiral alkyne, right, with 3 alcohols, 3 carbons with an alkyne, and then these 3 alcohols. And then they have these other, and then they have this other stuff, and they just start like adding these enzymes to it, right? And it's making these bonds, and it's putting it together, and then they have these auxiliary enzymes to like help it go forward. And believe it or not, at the end, islatravir just precipitates and they filter it. You know what I mean? Like, that's nuts.